All right, guys, thank you so much. We are doing a review for midterm 2, and we're going to go through lecture 6 to 10. All right? So, yeah, things are kind of going fast. So, lecture 6 was the heart dynamics. And before that, as usual, we go through exam format, section 1, multiple choice as usual. This time we've got 3 sections in the exam. So the 4 sections may show up again in midterm 3 and 4, and then the final 4. So we get, in this case, 35 times 1 equals 35 points. Section 2, our usual short answer section. We will have, you pick 5 of 6 times 2, which is the 10. And then section 3 will be the long answer. As usual, you have 4 of 5 times 5, which is 20, 30, plus 35, 65. So it's still worth 65 like last time, but this time you have 3 sections. All right? So here we go. So, electrical activity in the heart. All right. So, you know, we have to know action potentials, okay, because that's how the electrical activity is conducted in the heart. We all know about depolarization, but the cardiac muscle cells have 4 phases in the action potential. So if I were to draw it, it will be kind of like this. So you have the depolarization, early repolarization, and then you have your plateau phase, and then you have that. Okay? So no one's responsible for that. Of course, it's the sodium in as usual. All right? And potassium channels begin to sort of open up here for early depolarization, right? Okay. And then we have a plateau phase that's sustained by calcium ions, right? So note the 4 phases of the action potential in cardiac muscle cells and the ions involved, okay, for each of those phases, and you kind of be able to explain it. And then, of course, you know, up here sodium is closed, and then calcium is open, and then here the calcium, sodium are closed at the late or final repolarization, and then you have what? K plus channels opening, all of them now in this case, and then you get a rapid repolarization. All right, and of course the significance of that, you know, plateau phase is what? What is the significance of having that long plateau phase? What does it achieve for heart?
Resting.
Yeah, it gives it a more, a longer absolute refractory period, the time during which it will not respond to an action potential, okay? Because cardiac muscle cells are beating the whole time, okay? So there you go. So relative, we know that a stronger action potential can cause a stronger stimulus can cause an action potential, but you know, the absolute refractory period, which is anywhere from depolarization through all the repolarization, that's absolute. That means no action potential, regardless of how strong it is, will cause an action potential. So this whole deal is designed to rest cardiac muscles because the absolute period is longer. All right? So be able to explain those things about the action potential of cardiac muscles, right? Okay, and then the SA node, of course, is a group of cells that are autorhythmic in the heart. That means they have their own, you know, they can initiate their own action potentials, and therefore they set the pace of the heart. So it's, the SA node is actually the pacemaker, right? That's what we call the pacemaker. Okay. And again, there are different ions involved here. The action potential is like this. See what I'm saying? Whereas in the cardiac muscle cells, we had we had that plateau phase. In the SA node, we have the prepotential. Okay, we have sodium and calcium ions involved, but calcium ions are very much involved in the prepotential. Okay, that's very important. The prepotential delays what? What does the prepotential delay? Real quick. What is this phase of the action potential? Whenever you're on this side, what are you doing? What does prepotential achieve in the SA node cells? Yeah, but it delays the depolarization. That's what it does. The prepotential delays the depolarization, so that it will so that it means the length of the prepotential determines the pace of the beating of the heart, right? If you've got longer prepotential, the heart is beating slower. If you've got shorter prepotential, the heart is beating faster. Okay, so in order to regulate the beating of the heart, you regulate the length of the prepotential. Okay, so I want you to be able to explain those terms, the ions involved. Again, calcium is showing up here as the ion that's responsible for that. It seeps in ever so slowly, so that depolarization doesn't make that upward swing. It kind of slows it down, and then it depolarizes. It slows down, it depolarizes. Okay, so I'll be able to explain the action potential in SA node cells. The action potential in the rest of the cardiac muscle cells. Okay, because I will ask about that. And then, not only that, you have to also be able to tell me how the impulse is conducted throughout, right? So we're starting the pace with the action with the SA node, right? And then it goes through the atrial syncytium, which is the atrial, cardiac muscle cells, right? And then just before it gets to the AV node, it goes through what are called junctional fibers. Those are not mentioned in your book, but it goes through that, okay? Junctional fibers, which start to slow down the conduction of the action potential. You know what I'm saying? Okay, so it does that before it gets to the AV node, the atrioventricular node. When it gets to the atrioventricular node, it is also delayed, the action potential, to the tune of 0.11 seconds. So what are we trying to achieve by delaying this action potential? First with the junctional fibers, second with the AV node. What's the deal here? What are we trying to achieve? What's the purpose of all that? Why slow it down? Real quick, so that we can. Anybody want to hazard a guess? Why would you want to slow down?
To rest hard?
Not really this time. This time we're thinking in terms of cardiac cycle.
In terms of refill.
Yeah, you're kind of on the right track. The cardiac cycle is a sequence of events that must occur at a particular time in sequence, not together. So if you delay it, you ensure that the atria contracts and finish before the ventricles contract. So that's the significance. Because I will ask about those things. Okay, why do you slow down the action potential in cardiac muscle cells? What is the significance of the prepotential? You know what I'm saying? I'll ask about those things. So I'll ask you not only for you to tell me the information, but I'll ask you why is it, because we're kind of going up a little bit, you know what I'm saying? And then the delay of the conduction of the action potential through the cardiac muscle cell, what is the significance of that? Okay? Because you cannot have atria and ventricles contracted at the same time. So I will ask about that, guys, so be warned. Okay? So after the AV node, it goes into the AV bundle, sometimes called bundle of His, not His, bundle of His, that's his name. Okay? And then it will split into the left and right bundles. Now it's going fast because you've already delayed it a couple of times with the junctional fibers and with the AV node. Okay? And now it's going to spread through the ventricular myocardium using the Purkinje fibers. Okay? So I will also ask you through what structures the action potential is conducted in the heart. Okay? So gotta know action potentials in cardiac muscle cells, and the action potentials in the SA node, and how it's conducted, the structures through which it goes, okay, and the significance of that kind of conduction that occurs. Okay, so the electrocardiogram, which we'll be doing when we start the electrophysiology experiments, we'll actually measure this electrical activity, and we'll get our own P waves, our QRS complex. So the P wave is what? What is the P wave? What does it represent? P wave, the first one. So you're talking about the first chambers, the receiving chambers. So it's going to be what? What are the receiving chambers anyway? Okay, let's go back there. The receiving chambers?
Oh, the atria.
The atria. The atria. So it will be atrial depolarization, okay? All right. And therefore, atrial depolarization will precede what? What event does it precede, or does it come before? We measure the electrical activity in the heart so that because it causes a mechanical thing to happen. So that's what I'm talking about. What mechanical thing comes after atrial depolarization? It's simple, guys. The answer is in the
Atrial contraction.
yeah, atrial contraction. Okay, guys. Because all the reason why we are conducting electrical activity in the heart is to cause it to beat. Okay? So so I will need you to tell me that. Okay, what is a P wave represent, and what does it cause? What's the whole purpose of depolarizing atrial muscle? So that it contracts. Okay? So gotta know that, guys. Atrial depolarization of the QRS complex. What event is it? What does it represent? Yeah, ventricular depolarization, guys. Okay, ventricular depolarization, and of course it precedes, right? Ventricular systole, right? Contraction, or systole if you want. So, because I will need you to link this electrical activity to the mechanical actions of the heart. Okay, to the actual pumping action of the heart. You need to do that, and then of course the T wave is. What does the T wave represent? Yeah, ventricular repolarization. Okay, so we have covered all these three. Does it mean atria does not repolarize? Yeah, it must be the QRS complex. Okay, so know those things because I will ask about those things. Okay, guys. So usually in the cardiac cycle, when we say systole, we are referring to ventricular contraction. When we say diastole, we are referring to ventricular relaxation. Okay, and then when we actually qualify it and say atrial systole, then the atrial contraction. Atrial diastole, then the atria are relaxing. Okay, so I want you to link those cardiac cycle events with the ECG. Okay, like we are doing. What does the P wave represent? Okay, atrial depolarization. And what? How is that related to the mechanical action of the heart? It causes atrial contraction. Those kinds of things. Okay, because I will ask about that. Okay, and then of course the heart sounds. And then the first heart sounds, there's two of one, S1 and S2. Okay, S1 is due to what? What mechanical event is related to S1? Better go faster, guys. Okay, when the ventricles contract, they push blood up against atrioventricular valves, right? The tricuspid and the bicuspid. So when they shut, that's the first heart sound. Okay? Bye. You're shocking me now. Okay, S2 is due to what mechanical event? Okay, after the ventricles contract, where does the blood go? Where does the blood go? Yeah, so it's going to be that's going to be responsible for it goes through the pulmonary trunk and the aorta, right? When the ventricles contract. And both of those have semilunar valves. So when ventricles relax and blood tries to come back, it flops and closes, it fills those things, semilunar valve clasps, and you have the second heart sound. Because I will ask about that, guys. Especially now that you guys are kind of slow in answering now, I will definitely ask about that. You know how I do, you know. Aortic pressure curve is the, you know, when the ventricles contract, they are dumping blood into the aorta, right, which is the main artery, right? And therefore, it's going to have the most pressure during ventricular systole. So that's what we call systolic pressure. When you take blood pressure, the PA students were taking blood pressure the other day when we got the flu shots, and I got mine taken. So they measured my systolic pressure in my arteries, and they measured my diastolic pressure in my arteries when the ventricles are relaxed. Okay, so we know what kind of arteries the aorta is, right? What kind of artery is the aorta? It's very, what kind of quality? Elastic. Okay, it's very elastic that when you fill it up, it springs right back with so much force, it creates like a second heartbeat, which is called the diacrotic notch. Diacrotic literally means double beating. Okay, it looks like the heart is pumped again, or the ventricles have contracted again when the aorta recoils. Okay, so you can actually sort of feel like a second pulse. Okay, so know what the diacrotic notch is and what causes it. Okay. Also, the cardiac cycle, we need to know the events that are following the cardiac cycle. You know, like atrial systole, you know, of course, ventricular systole, and so forth. The atria contract at the same time. The ventricles contract at the same time. Okay, the blood pressure. Okay, again, I'm going slow right now. Okay, so I'm going to try and speed it up a little bit. But the first bit, you really have to understand all those action potentials and so forth. So this is pretty much talking about, you know, the stretching of the heart. That's what really it means, and how that affects the beating of the heart and the stroke volume. So if you're returning a whole bunch of blood to the heart, what's going to happen? It's going to stretch a lot. Okay? Just like a catapult. If you pull it a lot, I don't know, you guys don't use slingshots anymore. Okay? But we used to use slingshots. Okay? So, and you pull it, the more you pull it, that means you're going to send the pebble to a greater distance. So the more you stretch the cardiac muscle, the greater the force you're going to contract with. So that takes us to Starling's law. Okay? Which talks about the preload and how it affects the stroke volume. Okay? So greater preload or greater ventricular filling will result in greater force of contraction. Okay? So that's what it is. Right? So the Starling's law. And there are intrinsic mechanisms that the heart controls itself or self-regulates. After all, it is autorhythmic, so it sets its own pace. So there are ways that it controls itself. Okay. One of the ways is that if you have a lot of venous return, it will contract with more force to push the blood out. All right? So that it will limit the end diastolic volume. You know what I'm saying? You see what I'm saying? Because if you contract with greater force, you squeeze more blood out of the ventricles. And therefore the volume left in there is less, which is end diastolic volume. End systolic volume, I'm sorry. End systolic volume, because after it contracts systole. End systolic volume is much less when you contract with greater force, okay? So now there's the concept of afterload, okay? What is afterload? This is the force that the heart has to beat against, depending on what the force of what's already in your arteries. So if you've got high blood pressure, your lower diastolic pressure is much higher already. Okay, maybe because you know your muscles are contracting in your smooth muscle in your arteries or whatever, or they're getting more rigid as you get older. So it gets higher pressure in there. So the afterload is the force that the heart has to beat with in order to oppose that force that's already existing in the arteries. That's why you need to lower your blood pressure, because the higher your blood pressure is, the greater the work. So the afterload will be much greater for the heart, right? And therefore the heart has to really beat hard just to force the blood through the aorta. See what I'm saying? So that's the afterload, okay? Know what the preload is, know what the afterload is, and know the significance of you know, the existing pressure in arteries. Starling's law, be able to explain it and how it is related to preload, which is greater filling, right? And how it's related to stroke volume, which is much greater stroke volume, because it will contract with more force, it forces more blood out. Which means the stroke volume increases by decreasing end systolic volume, right? Because end systolic volume is much less. What is left in the extrinsic regulation? Now, this will be hormones, in the nervous system. Okay, there are certain hormones like even atrial natriuretic peptide, which is produced in the kidneys and comes because the natriuretic means sodium. Getting rid of sodium also gets rid of water. Retention of sodium also retains water. The more water retention, the greater the blood volume, the more the pressure, right? So that's why you don't want to eat too much salt, especially when you get older. Okay, so those are the extrinsic ways, something outside of the heart controlling it. Okay, like the nervous system, we're using the cardiovascular center, cardioregulatory center in the medulla oblongata. Okay, it is sensitive to the blood pressure. Okay, and there are baroreceptors also in the aorta, the aortic bodies, and the carotid bodies. It is detection of pressure. So if pressure goes up, it goes to the cardioregulatory center, and then it will send what kind of nervous stimulation to the heart. If blood pressure goes up, what kind of stimulation is sent to the heart? Parasympathetic or sympathetic? What slows down? I said if the blood pressure goes up and the cardiorregulatory center senses it in the medulla oblongata, what kind of stimuli is sent to the heart? Is it sympathetic or parasympathetic? The pressure has gone up. What does the sympathetic normally do? Normally stimulates, right? So you send parasympathetic stimuli through the vagus nerve. Okay? Because the parasympathetic normally decreases, normally inhibits. Okay? So I'm going to need you to be able to tell me those things. Okay, so that's extrinsic regulation, okay? The parasympathetic normally decreases, okay? And the sympathetic is fight, flight, normally stimulates. So sympathetic will come in if the pressure detected by the cardioregulatory center goes down. So you send through cardiac nerves sympathetic stimuli to speed up heartbeat, okay? So be able to explain these intrinsic and extrinsic methods of controlling the heart, okay? Okay, so please go. I need to pick up the pace here. So you also need to tell me how, we talked about the effect of blood pressure right now and the cardiorregulatory center in the medulla oblongata, but there's also the effect of pH. pH is related to what mostly? That's in blood. pH is mostly related to carbon dioxide. Okay, so that will affect more the respiratory system, you know what I'm saying? Okay. So these chemical factors affect more the. Why does these chemical factors affect more the respiratory center rather than speed up heart rate and so forth? Cardiovascular center.
Well, that's what deals with your lungs.
Yeah. So why affect more the stuff that deals with the lungs instead of the stuff that deals with the heart? When you talk about pH, CO2, and O2, the answer is right in there. What deals with the CO2, pH, and O2? Yeah. So no matter how fast your heart is beating, if you're lacking oxygen, it's not going to happen. You're just going to pump the blood fast and you're not going to get any oxygen. So those factors will affect more the chemoregulatory centers will affect more the respiratory center because it's talking about the oxygen and the CO2, which is regulated more by the respiratory center than by the cardiovascular center. Okay? The pressure affects more the heart, the cardiovascular center. Okay? Because, it's the heart that's doing all the pumping. Okay? Now, the effect of ions and body temperature, we know about calcium, potassium. It will follow. Oh, I should have done the superscript here, but I forgot. I'm going fast. Okay. If, let's say, you have high extracellular potassium ions, what happens? When you have high, how does it affect the cardiac cells? I'll only talk about that, and then the other ones you can figure it out because, you know. Well, I'm talking in terms of, you know, think about when you have ions outside and the charge they're carrying. So if you have more of those ions outside, it means they're going to move in, right? Because of concentration gradient. So if you've got potassium ions which are positively charged outside, and they start to get into the cell because they're on the concentration gradient. So if you're getting a positive charge into the cell, what are you doing? Yeah, you partially depolarize. Okay? So when you have high extracellular potassium, it gets into the cell, you're partially depolarizing. So that the amplitude of the action potential, you see, this is the action potential, right? Depolarization, repolarization. The amplitude of the action potential will be less. So let's say you've partially depolarized up to here. When it finally depolarizes, this is only going to be the amplitude. Also, from here to there. You know, I'm trying to do double thing easy, just sense that. When you reduce the amplitude, you also reduce the force of contraction, so you affect stroke volume and the rate of the beating. Because the amplitude of the action potential is lower. Okay? So that's what increased extracellular potassium does. Okay? Read that section I told you to read in the book because it will explain all that. Okay? So all you need to know about this is the action potential and what happens. You know depolarization, you know repolarization. So if you have an ion, what matters is the charge of the ion, not really the ion itself. Okay? So wherever you have more of the ion, it's gonna go wherever it's less of. So, and you know the effect of that. If you get a positive charge into the cell, you are, partially depolarizing. And if we have more intracellular, potassium, what happens then? It moves out. And then when a positive charge is moving out, so that it's becoming more negative inside, you are hyperpolarizing. So it will affect the heart rate, but it won't affect the stroke volume. Okay, so things like that. So read that section. Okay, temperature, of course, the higher the temperature, the faster the heart beats because it will get rid of the temperature more. Okay, so and then calcium, high calcium affects nerve impulse stimulation, so it will slow down heart rate. Okay, but low calcium, they have to go to really low levels, more than 10 times low, for them to be able to affect the heart. Okay, when it goes on the loss. So effect of aging, when you get older, of course, your heart becomes less elastic, and then it starts to have more preload, afterload, and it starts to grow hypertrophy and so on. Okay, I spent a little bit too much time on that one, so this one is easier, sort of, okay? But with the heart and all the electrical activity in the heart, I really want you to understand that. And all those actions and how they are related to the mechanical, those action potential. So the lymphatic system has got two systems, right? That are kind of sort of interdependent, a little semi-dependent. Okay, the network of vessels, of course, which are linked to the lymph nodes, all those things, tonsils and whatnot, which are the organs or the lymphoid tissues. All right. So those are the two, and then of course the functions. The functions of the lymphatic system, right? Draining excess fluids, returning proteins to blood, and so forth. Okay. So know those, and then lymphatic vessels, capillaries. And then you get to the larger vessels, which are called lymphatic vessels, which lead to the lymph nodes, and then the ducts. What are the two ducts? The big main ducts that dump into the subclavian, the thoracic and the thoracic duct and what's the other one?
Right.
Yeah, right, lymphatic duct. Okay, guys. All right, very good. Okay, so. Gotta know those guys, and then the right lymphatic duct, okay? Those are the major ducts. So know the characteristics of the vessels, okay? The vessels are similar to what other vessels of the body? The lymphatic vessels are similar to what? Starts with a V. Veins, yeah. Veins, they got valves and so forth, okay? The movement of lymph also relies on the same actions that move blood in the veins. Okay, so veins and lymph lymphatic vessels are the same. Okay, so again, those three things know what causes lymph movement: vessels, smooth muscles, okay, and then of course the unidirectional flow with the valves, and then of course the respiratory, contraction and relaxation, or expansion and contraction of the thoracic cavity as a suction and the pressure thing. So gotta know that, and then of course when skeletal muscles contract, what happens? They squeeze up against those lymphatic vessels, just like it does the veins, and then blood is squeezed up, and it only goes up because the valves will ensure that it goes only in one direction, right? Okay, so no characteristics like the valves and so forth. They are blind-ended tubes, but the endothelial cells overlap, so they are able to open depending on where the pressure is greatest. Okay, and they shut when the pressure increases inside of the lymphatic vessel. What is lymph then? What is lymph? What is lymph? If I ask you the exam to define lymph, what is it? It's fluid that. What kind of fluid is it?
Also water.
Well, I'm talking in terms of name of it. What's the name of it? What's the name of that water you're talking about, that fluid? Interstitial fluid, right? Okay. So it's interstitial fluid that has entered a lymphatic vessel. That's what lymph is, simple. Okay. So you gotta go through your notes, guys. All right. The lymphatic organs, okay, tissues and so forth. We've got the diffuse lymphatic organs like the tonsils, lymph nodules, lymphatic nodules, and so forth that have no connective tissue covering. And then we've got the actual organs like the lymph nodes, of which there's many, like there's almost 500 of them in the body. And then the spleen and the thymus, right? So, and then, of course, some of this diffuse lymphatic tissue is strategically located, such as the tonsils around your throat, nose area there, anything where you have a mucous membrane taking in stuff where there's a risk of infection, we have that lymphoid tissue. And you also in the pia pages in your small intestine, the distal part of your small intestine going towards the appendix, where which is actually breeding the bugs that are cohabitating in your the gut microflora. All right. What are the crypts on these tonsils? What are they? Remember crypts?
Little pockets.
Yeah, little pockets that trap and then destroy the
Increased
bacteria.
surface area.
And increased surface area because it's almost like the same as microvilli and so forth. Okay, guys? And then there's three, of course, palatine, lingual, and pharyngeal on the back of the throat, lingual under just at the base of the tongue there, and the palatine on the sides. Okay?
So the ones you look at when you look into somebody's throat or the tongue.
Yeah, palatine ones are the most, especially when they're inflamed and they're all swollen up and you can see them going up. Okay? So lymph nodes, we said they are the most numerous, okay? And we said, and then there's ones that are superficial. Where are the superficial lymph nodes located again? The three areas? Yeah, so we call it cervical. Okay. If you say that, I might give you the points and look away. But you gotta say cervical, which is the neck, and armpits will be the axillary. I know. Okay, thank you for trying to speed it up, though. And then, of course, the functions. What is the functions of lymph nodes? One is to filter, right? Filter. And then what's the other one? Yeah, to activate the immune system, because that's the whole point. They're the ones that see it first, and then they shout out, "Hey guys, we got some visitors here that are not welcome."
Destroy lymphocytes.
Yeah, destroy. Because how the helper cells are activated and whatnot. So it activates the immune system, okay? And then, of course, the characteristics, you got that, the 4 layers, these lymphatic sinuses, the general center, and things like that, lymphoid tissues, lymph nodules, okay? And then you got the medulla and the cortex on the outside, okay, right? And you got those trabeculae that are formed by, the sort of invaginations of the connective tissue covering that divides it into what? Sections. So know those characteristics, know functions, and also where those superficial are located, and everything else is deep. You don't feel it. The rest of it is deep. Lymphatic organs, So, you know, the spleen is in the left upper quadrant of the abdominal cavity, you know. So here I need you to know about the white pulp, okay? And then the red pulp, which is associated with the veins, and this is associated with arteries, okay? And the white pulp has lymphatic lymph nodules. Yeah, red pulp is associated with the veins, okay? And it's got reticular sort of tissue in it, and then, well, it's got reticular cells that are sort of star-shaped cells. And then this is associated with arteries, because when the artery, the splenic artery comes in, it goes into the what? And it splits into capillaries, goes through the white pulp, okay? So know also, you know, it acts as a small blood reservoir. The major blood reservoir, of course, in the body is the veins, and then, of course, followed by the liver. But this acts as a sort of minor blood reservoir, okay? So you need to know blood movement in the spleen, right? As it comes through the splenic vein, branches into the arterial, and so forth, right? Goes into the capillaries, right? And how it goes straight into the venous sinuses or diffuses through a gap, you know, and so forth, or goes into the red pulp, into the you know, red pulp, the reticular fibers there, and then sort of filters into the venous sinuses. So know about the movement of blood in there, in the spleen, okay? Okay. The thymus is bilobed, of course, and it secretes hormones, thymosin and the thymopoietin, right? Okay, and those are meant for what? What are those, the thymosins and thymopoietin meant for? They are supposed to mature which cells? T cells, right? Okay. So this is an unusual lymphatic organ in that it doesn't actually filter or attack the microbes. It just pretty much generates T cells. It matures T cells, and then the T cells will migrate to other tissues where they do the killing, okay? But not in the thing, in this thymus, okay? Its sole purpose is to mature T cells, and that's it, okay? And also, is the one that is those are you know, star-shaped cells, reticular cells, instead of having reticular fibers like all the other lymphatic organs have. Okay, so those differences. Look in your notes, you'll find them. So decreases size with the age, of course. When you're older, you've been exposed, and you don't need a whole bunch of T cells like you do when you're young and naive and not exposed. Okay, so know those characteristics. So lecture 8. So blood. So we need to know all the composition of blood and all the characteristics of those and functions. All right. So blood maintains homeostasis really. Even temperature-wise, it's involved because it can release temperature, and all the chemical components of blood are homeostatically controlled. So it's involved in that. And then, of course, all the other functions like moving nutrients, right? Gases, oxygen, carbon dioxide, what else? You know, transporting molecules, processed molecules, and things like that, right? So you need to know all those functions, okay, guys? And be able to kind of sort of talk about it a little bit. And then, of course, blood is connective tissue, right? Because it's produced where? In red bone marrow, which is the epitome of connective tissue. All right. So blood is the only fluid, matrix in the body because of the plasma, and it has the formed elements. What are the formed elements in the blood? What? Formed what's that? Red blood cells. What else? White blood cells. What else? Platelets, right? Platelets are not true cells, but they're still formed elements. Okay, they're parts of cells. So we need to know what is hematocrit.
Percent of red blood cells in the body.
Yeah, in whole blood is a percentage of whole blood. Yes. So characteristics of blood. It's kind of this metallic tasting sticky fluid. It's red and so forth. All right. You go. Plasma. Plasma, you know, it's a sort of straw-colored, foam colored, and it, has proteins in it, you know, and so forth, plasma proteins. The major of which is albumin, you know, which is a function of albumin is osmotic pressure, right? And then immunoglobulins, which we'll talk about in a little bit, which are involved in immunity. Okay. So gotta know that. And then fibrinogen, which is involved where? What is fibrinogen involved in? What's that protein do?
Body.
Yeah, protein. So that we talk about protein and other substances such as the ions, the electrolytes, and all those things, okay? And waste, urea, things like that, okay? All of which are homeostatically controlled, okay? So gotta know all that about blood. And then, of course, the formed elements. Those are red blood cells, white blood cells, all right? And I need you to know about the life cycle of red blood cells, you know, how they start off from the hemocytoblasts and then pro-erythroblasts, right? Early pro-erythroblasts and how it starts to form ribosomes and, you know, and so forth in the early pro-erythroblast, and then it loses, you know, the ribosomes and whatnot, and late erythroblast then loses the late pro-erythroblast loses the nucleus and becomes a reticulocyte. Finally, then it will mature into the red blood cell. Okay? So know about that. And then, of course, white blood cells, we got two major types, cycle and you know, development, okay, in terms of from the hemocytoblasts. Okay, white blood cells, what are the two major groups of white blood cells?
Mononuclear and poly.
No, I'm talking about the major overarching groups. Are granulocytes?
Wouldn't have got that.
Why did you shout it out? Monocytes
I
and
wouldn't have got that.
lymphocytes. Oh, you wouldn't. Okay, and then the other one, of course, is the
Granulocytes.
yeah, granulocytes. These ones have granules, and we got the neutrophils.
Basophils. Eosinophils.
Okay, this is your turf then, you know what I'm saying? In terms of identifying them and whatnot, okay? Eosinophils, which is good. Eosinophils, okay? And they just differ in the acid-base stain. These are neutrophils stained purple because they are stained in both acid and basic stain, all right? And these will stain blue, dark blue, black in basic stain, basophils will, all right? And then eosinophils, of course, will stain red in acid stain, you know, so forth, okay? And then, of course, you know, how they are involved in disease, like neutrophils, bacterial infections, and so forth. Eosinophils, web parasitic infestations, and things like that. So when you do a complete blood count, you can tell, you can have the diagnostic thing, which we'll talk about at the end, right? Because then if you have more neutrophils, then you say, oh, the person is infected by bacteria. You give them an antibiotic and they go, right? And then if they've got raised eosinophils, you might say, oh, there's a parasitic infection, then you do that, all right? So platelets are involved in the clotting, and we'll talk about that. And they arise from megakaryocytes, okay? They're pieces of those big cells. Most of the formed elements, okay, survive for a few days, okay? Only the red blood cells last for about 120 days, which is like 4 months, you know? So every third, you get new red blood cells in your blood, okay? You know, the function of red blood cells, okay? And, you know, in terms of, you know, transporting oxygen and carbon dioxide, and hemoglobin, then we need to know about hemoglobin and all that, okay? Growth factors determine what kind of cells will arise from those hemocytoblasts. You have those one stem cell that forms all the blood cells, okay? So you've got the pro-erythroblast, you got the megakaryoblast for the megakaryocytes and so forth, myeloblast and lymphoblasts. For the lymphoblasts will be the granulocytes, and the myeloblasts will be the granulocytes, right? The neutrophils and whatnot. Okay, so know about how that affects hematopoiesis, of course, formation of blood cells, and it differs between the fetus and the adult, because in the fetus, you're making a new individual all from scratch, so it's made in all the organs, liver, whatnot, and all that, okay? Whereas when you're an adult, it's limited to red bone marrow, which is limited to certain bones even. Okay, not all the bones have red bone marrow. Most of the bones have yellow bone marrow. Okay, which it starts to get replaced as you get older with yellow bone marrow, the red bone marrow. Okay, so red blood cells, of course, you can know the characteristics. Of course, you got hemoglobin, they are biconcave and whatnot. And hemoglobin, the shape of the red blood cells is really the epitome of relation of characteristics to function. Because the shape of it gives it a very tiny center, and they can squeeze through and do all sorts of stuff. Okay, because of that biconcave discs, okay, they contain a whole bunch of hemoglobin, in fact, a third of the cell's hemoglobin, okay, which is really not a cell at this time because it has lost most of the organelles, and all it has is the hemoglobin, right? Okay, then know about hemoglobin in terms of how it has those 4 heme groups, okay, how it's recycled, okay, the breakdown of hemoglobin, where you have the globin and the heme is broken down to iron and the pigment bilirubin, right? And how the bilirubin goes to the small intestine via the liver through the bile, and then it's digested in the derivatives of bilirubin, which then give the brown color of feces, and some of it is reabsorbed, goes into the blood, and then goes to the kidneys, where it gives the yellow color of urine. Okay, so gotta know about all that cycle, and then of course gotta know about how hemoglobin transports oxygen. There's only 2 ways that oxygen is transported. Okay, attached to hemoglobin is about 98% of it, which is obviously hemoglobin, right? And the other one is, how is oxygen transported?
Plasma?
Yeah, it's dissolved in plasma. Okay, but carbon dioxide is 3. Okay, what are those 3 ways that carbon dioxide is transported? Right? Okay, we got dissolved in plasma, right? It's carbonic acid, which means it goes to bicarbonate ions also, and then attached to hemoglobin, right? To the amino group, to the globin part, okay, not the heme part. That's for oxygen. Okay, so it forms carbaminohemoglobin. Okay, so those are the three ways. The most ways is transported is bicarbonate ions. Because when CO2 is dissolved in water, it forms carbonic acid, and then carbonic acid dissociates to H+, which is why carbon dioxide is related to pH, because it will form H+ and HCO3, which is the bicarbonate ion. The most form. So gotta know the 3 ways that this is transported and the 2 ways that that's transported. Okay. All right. What? It stopped mid-action. And then, of course, red blood cells, erythropoiesis, the formation of, so we talked about it a little bit, I referred to it. Hemocytoblast becomes a pro-erythroblast, and then an erythroblast, and then, starts to make a bunch of ribosomes and stuff because it makes the protein hemoglobin. And then late, it does its thing, it loses the ribosomes and so forth, and then loses the nucleus and becomes reticulocyte. All right. So I want you to know that cycle, and then it will mature into a red blood cell after that. Okay. Hormonally controlled, by erythropoietin, EPO, right? When your red blood cells go down, you release erythropoietin from the kidneys, right? Because the kidneys are filtering blood, they know what's going on. Okay, so, and then you need vitamin B. Vitamin B is like the folate is a vitamin B, B12. You need that. And iron, of course. All right, lifespan we talked about around 20, and then we need to know about hemoglobin breakdown, which I said, how you go into the globin and the heme, and then the iron is recycled. The heme becomes bilirubin. The globin is broken down to. What is the globin broken down to? Real quick. What is globin anyway? What is globin? What is it? Is it a carbohydrate? It's a protein. So what do you break it down to? Amino acids. Okay? And those are also recycled in this. Okay? So know about hemoglobin breakdown, the cycle of red blood cells. Okay? So now we go to white blood cells. Those are the only complete cells. The rest of the red blood cells, we call them that, but they don't even have a nucleus. They've lost all the organelles, okay? So they're not really cells if we're being strict, okay? So those are the only complete cells, the white blood cells are. Okay, and the functions of them, of course, mostly function in the immune system, okay? And you got neutrophils, which are macrophages, and then, monocytes and macrophages. So, like we said, we've already split these, I already preempted, so we're not going to dwell on that. We know our granulocytes, and we know our granulocytes, right? Those three neutrophils, basophils, and eosinophils are granulocytes, and then the other granulocytes and monocytes, lymphocytes. Megakaryocytes, now we're going to the clotting part. Okay, so the two overarching clotting mechanisms is a platelet plug. Platelet plug is meant to repair the small cracks in your blood vessels that are going on thousands of times during the day. Okay, otherwise, if it wasn't a platelet plug, you'd go bled out inside, which is not good. Okay, so that's for small little cracks, that's nicks that happen on our, capillaries. Platelet plug, and clots are for the big ones, like big major cuts and whatnot. Okay. So if I ask about the just two clotting mechanisms and their functions, I'll say name two clotting mechanisms and their overall functions. Okay. Then you talk about the platelet plug, which fixes the small nicks and cracks that happen in our capillaries. Okay. And then of course the clots, which fix much bigger injuries. Okay. Of blood vessels. Okay, so stoppage of bleeding, there are three ways. Vascular spasm, what is a vascular spasm? Contraction, smooth muscle, right? And therefore it will shrink. Sometimes the smaller capillaries can completely shut down after the vascular spasm. Okay, and then of course the platelet plug formation, which happens when collagen is exposed, right? And you get von Willebrand factor from the epithelial cells that will stick those platelets onto the collagen to kind of cover it up and start to form a plug, all right? And then you have the platelets, they're activated by ATP, adenosine diphosphate, right? And what else? And thromboxane, right? Okay, so know about that, how the platelet plug is formed, and then they start to get those fibrinogen receptors on the platelets which are attached to each other, and then they form a plug. And then of course coagulation, which is clotting. Okay, it has two ways. Intrinsic mechanism is caused by something in blood, which is called engagement factor, right? Factor 12, X11. Okay, and then the extrinsic is caused by. What is the extrinsic caused by? It's something outside of blood that is released when tissues are damaged. What is it? Thromboplastin, right? So that's when tissues are damaged, they release that, and then so the platelets start to become sticky and whatnot, and then they form a clot. Okay, so and then of course the overall mechanism, the extrinsic way or the intrinsic way, they all converge at factor 10, right? And then factor 10 will then activate together with phosphates, and all the other factors will form prothrombin, thrombin from prothrombin will convert that, and then the cascade moves on, and so forth. So know that cascade, okay, and how it starts off at factor 12, okay, and then at the end you get what is called clot retraction, which is what happens. They contract those platelets so that they pull the edges of the wounds closer together, okay, and then they release. What fluid is released when that happens? What fluid is released when the clot retracts? It's similar to plasma, but it's called serum, because serum doesn't have clotting factors because they've been used up in the clot. Okay, so know about that. Okay. Okay, and know the things that control, like platelet plugs are controlled by prostacyclin, which sort of dissolves them, and then the clots are controlled by the tissue plasminogen activator, which activates what? Plasminogen to become plasmin. So plasmin, starts to break down those clots. Okay. fibrinolysis. So any of these mechanisms of clotting have a control mechanism because you don't want it to just happen uncontrollably. 9. Okay, we're doing not so badly, but we need to speed it up a little. Okay. So immunity. There's two big groups: innate and adaptive, right? So the first one was on the innate, right, which are just is natural, non-specific mechanisms, just like engulfing microorganisms, you know, with macrophages and whatnot. Phagocytosis is non-specific; it will engulf anything foreign and then break it up. Okay, and then what else? Inflammation,
Fever.
yeah, fever, you know, which are stimulated by pathogens, and then it goes to the chemicals that you need to know. Okay, the two major chemicals, apart from pathogens, kinins, and all those, and cytokines, is complement, which is a group of about 20 or so proteins in the blood that will, you know, lyse cells. Okay, and you know, complement can be activated, you know, via the classic pathway, right, which is attachment to antibody antigen complexes. Remember, on the constant chain, there's a part where the complement proteins attach, and then you know you go to complement fixation, and then it will kill those cells that are infected. And then the other way, the alternative pathway, okay, which is the innate stuff. Alternative pathway is the innate, and this is the adaptive pathway of activating complement. Okay, so that's why complement is one of the proteins or one of the chemicals that are involved in the innate immunity. Okay, via that alternative pathway. Okay, so that's with factors B, T, and P, and you know, it will activate C3, which is complement protein number 3. Okay, and then you get that complement activation. And then the cells, chemotaxis. Okay, those white blood cells are attracted by chemicals that are released by, you know, the tissue that's infected, and so they're attracted to go there. Or when the tissue is damaged, it will release some of those factors. Some of them cause the clotting, like thromboplastin. Some of them will attract these white blood cells, right, via chemotaxis. And then also these white blood cells can squeeze out of and you know, the capillaries via those things, by the process called diapedesis. Okay, diapedesis. That's when it's kind of like an amoeboid action, and they kind of sort of squeeze through the gaps between the endothelial cells. The gaps get bigger though when during inflammation, it opens up them more so that white blood cells can get out. Because white blood cells are, after all, complete cells. They got everything in there. They're all packed up. Okay, so that process when they squeeze out is diapedesis, right? And then of course inflammatory response is a non-specific way. What does inflammation achieve? What is inflammation anyway? What is it doing? What's happening? What is inflammation? Yeah. And so white blood cells are contained in blood. Yeah. So inflammation is rushing blood to the area. That's what it is.
Dilating.
Yeah, and dilating blood vessels so that these guys can squeeze out the white blood cells. So inflammation, that's why when you're inflamed, it becomes red, because there's a bunch of blood there. Okay? And then,
Hotter.
yeah, it's harder, because, you know, the blood comes with the heat. So that's what inflammation is. If you bring a whole bunch of blood, you're going to, of course, bring a whole bunch of everything there. White blood cells, even red blood cells, because you need oxygen to, all the energy that's required to break down everything. So inflammation is simply dumping a whole bunch of blood so that the cells go to work. White blood cells do their thing, red blood cells do their thing. And then the clotting, if it needs to happen, will happen when the guidance. Okay? That's why even when you get a cut, that part becomes inflamed too. Because blood comes rushes, it becomes red. Same thing. So the inflammatory response is one of the ways. And then, of course, adaptive immunity. I want you to know some of the other chemicals involved, though. We already mentioned pathogens from chem, right? And then the kinins, of course, and then the complement we mentioned it. Interference is another one. It's a big one. This is really activates deactivates viruses. And they are, I think, treating some of like Ebola virus with some of interference. I think maybe, you know, they tried it with AIDS, HIV interference. It was successful, and then for a while this quit. I don't know what happened to that research. They were doing it in Kenya. Anyway, but interferons, they kill viruses or virus-infected cells. Okay, so adaptive immunity is specific, and we'll talk about it in the next lecture, right? That's the last lecture. Okay, and here we also talked about blood grouping, right? In the same lecture as immune system 1. And then you see we get these sort of glycoprotein antigens, which are on the surface of red blood cells, okay, that are the basis of the ABO blood grouping, okay, based on their presence on those cells. So you've got A antigens in blood type A. So what kind of antibodies do you have in your system? If you have blood type A and you've got A antigens when you have blood cells.
B.
You have B, because if you have A, you're going to agglutinate your own red blood cells. That would suck, and that would be bad. So, you see what I'm saying? So, I want you to know, of course, the antigen and what the antibody is, okay? And then, transfusion reactions, of course, occur with those immunoglobulins on the B cells, okay? Those are the ones that are immunoglobulin M is responsible for the transfusion reactions. Okay, and then of course the ABO blood grouping. Okay, so there I just want you to know the antigen that is there and the antibody that's there, and then of course understand which blood group is the universal donor, which is O, because O doesn't have any A or B antigens on the red blood cell. It's just like that, and there's the biconjugate. There's nothing. Okay. At least for the ABO, it doesn't mean it's completely devoid of antigens on it. Okay, and then we talk about the rhesus, which is antigen D. It's another type of antigen that is also found on red blood cells. The only reason it's called rhesus is because it was first discovered in rhesus. Because those are our primate friends that when we do research, we go there.
So that happens to monkeys too?
Yeah, they have the same. Yeah.
They
have all those things. Yeah. Because they're just primates, just like us. Yeah, they have those things too. Okay, but nature is a way of correcting that, because those guys, nobody's watching their kids to see if the mom is negative or not. You know what I'm saying? So, but nature controls that, because they're in the wild. They're supposed to be, and we kept it.
No Rogaine for that.
What's that?
No Rogaine.
No Rogaine.
So you said Rogaine.
I did too.
Me too. I thought you said Rogaine, and I was like, are they bold or what? Like.
I don't
Okay.
think monkeys suffer from male pattern baldness.
They might actually. The male ones, they might, because they are our friends, they are our close relatives. Hemolytic disease of the newborn is caused by that IgG, which will cross the placenta, and it only happens if the mom is negative. Because when the mom is negative, it means it will have antibodies for the rhesus factor, for the rhesus antigen. And if the baby is positive, really? So whenever she goes, they will.
My grandma was still male and was born really sick.
Yeah.
Blood transfusion. So they didn't know.
They didn't know about it. Now they know about it. So all the women who are negative, they don't even bother. They treat them prior to pregnancy,
Right
yeah.
after.
Yeah, and after too, because when they breastfeed, especially because they will have all those antibodies in there and colostrum in the milk. So hemolytic disease of the newborn happens because of that, and the rhesus factor, and the mom will start to become sensitized and produce the antibodies for the antigen D, which will start to kill the cells in the fetus. So it happens on the second time. The first time she's just sensitized. The second pregnancy, if it happens to be again positive, which might be because the husband is probably the same husband, you know what I'm saying? So yeah. So and then then you're prompt, because the mom has got antibodies now waiting and sitting in there. As soon as that fetus starts to grow, boom, it gets attacked. All the blood gets attacked. Okay, so diagnostic blood tests, of course, are used for to determine a lot of things. Like if you look at under the scope, you can see the shape of the red blood cells, that you can see sickle cell anemia and so forth. And then the type and number of white blood cells, we talked about it just now. We said eosinophils, parasitic infestations, neutrophils, bacterial. So if you see the neutrophils elevated, then you know it's a bacterial infection, you give them antibiotic. So that's what we mean. So just know what things contribute to the diagnostic use of blood. Okay? And then, of course, chemical. we can do a chemical analysis of the blood. We see a bunch of urea in there and all that. We know that the kidneys are failing. Okay, because of the ones that get rid of that. Okay, so you know, complete blood count, of course, that's what you do. So you know what's in there, the white blood cells and so forth, and then you can be able to predict what kind of bug. And hematocrit, of course, if your hematocrit is low, then you're probably anemic, you know, due to many kind of causes. But the end result is the same. You are lethargic, you know, you don't have oxygen and whatnot. And then clotting, sometimes you don't clot. I happen to have low platelet count, but sometimes I cut myself shaving the other time, it wouldn't stop. And I was like, oh, it must be the low count of my platelet. For some reason, they're just low. Okay? But they measure prothrombin time, which is the time it will take your blood to clot the prothrombin, and they see how it's converted to thrombin. The thrombin activates the clotting cascade, right? Okay, so last lecture, guys, and we'll kind of breeze through this one. Now, the difference between innate and you know, adaptive is that adaptive is specific and adaptive has memory because it creates those memory cells. Okay, so it destroys the foreign antigens. It amplifies inflammation. Again, we know what inflammation is. It just dumps a whole bunch of blood in there, so it brings all those cells with it. And then activation of complement, of course. Remember, we talked about it on the chain, that constant region. There's a part where complement attaches, and you know, you get complement fixation, and macrophages will attack that. And then, of course, the two divisions. We got antibody mediated from the B cells and the plasma cells that form a whole bunch of antibodies specific to that organism, and then cell mediated. We will start using the cytotoxic T cells. Okay, so know those two branches. And then, of course, antigens. What are they? These are very light molecules, up to the tune of 10,000 molecular weight. That will elicit an immune response from the adaptive system, right? So we know about the origin of B and T cells like bone and thymus, and we know about positive selection, where you select for the ones that you know, will attack foreign microorganisms, and negative selection, you select against those ones that will attack your body proteins. So they are attached to host epitopes. They're your own epitopes. So the epitopes are the parts of the antigens that the antibodies attach, okay? So MHC class 1, this is for an internal antigen. Remember how that's processed through the endoplasmic reticulum, Golgi body, and all that. So this will mean the cell is destroyed, okay? Because it's infected. When it's a professional antigen-presenting cell, then it will exhibit the antigen on class 2, MHC class 2, which means it will stimulate the immune system. That cell is not killed, because it's one of the guys, right? But if any cell becomes infected, even if it's an antigen-presenting cell, if it's a dendritic cell, it doesn't matter. It will be cytotoxically killed. Okay? So that it will, because it will show the bug on the MHC class 1. And MHC class 1 antigen complex, it means cytotoxic T cells will kill that cell because it's infected. Okay? So know the difference between these two, 1 and 2, and then of course helper T cells where they're activated. Okay? They attached to the MHC class 2, usually coming from an antigen-presenting cell professional, and then you activate cytokines, it brings cytokines, because it will activate other parts of the immune system to kill the bug, so that the cell is not really killed. So these activate by producing cytokines that will, and then B cell activation, of course the intention will be to then form the plasma cells, which will form a bunch of antibodies, right? Okay, so know that, and then of course tolerance. You suppress the T cells. The tolerance is what's responsible for us sparing our own cells, right? Okay, immunotolerance. When you have problems with that, you probably let some B cells and and so forth survive that are attached to host epitopes, and you have an autoimmune situation. Okay, lupus. Yeah, lupus is an autoimmune disease. It attacks your own body cells. Okay, and then of course I need you to know these immunoglobulins, right? We just talked about IgG, which crosses the placenta and it causes the hemolytic disease of the newborn. Also activates complement. This one also acts as an antigen receptor on B cells and is one that's responsible for amio transfusion reactions. All right, AGA is in saliva. It's in all those surface chemicals, tears and whatnot, to protect mucus membranes, okay, anything that takes stuff from the outside, like the respiratory system, the digestive system. So that's that. And the IgE is on mast cells and basophils, and it stimulates an inflammatory response during allergic reactions, okay, like hives and so forth. And then IgD is also acts as an antigen, receptor on the B cells, okay? So I want you to know this. Sometimes I'll ask, can you give me the 5 classes on antibodies and what their function is, okay? I will do that. Effect of antibodies on antigens, right? Agglutination, and just destabilizing them, and then, of course, complement. So there are 5 ways that happens. And also, it will activate macrophage and would start to attack it, okay? So those ways that antibodies disable antigens, gotta know them, that's 5, okay? So gotta know those. And then primary and secondary response, we know that the primary response is body still naive, right? And then when the secondary response, you get a whole bunch of memory cells now. It's more robust, okay? It's got no chance. That's why you only get a flu strain only once, because the second time it comes, you're ready. Cell-mediated immunity, those are the cytotoxic T cells, and of course, this will be anything presented on the MHC class 2 will be killed. Okay, that cell will be killed because it will be infected. Okay, and then acquired and adaptive immunity. We know about that. We get the active and the passive. Okay, the active, you're naturally getting infected with a live bug and you get sick, but you build antibodies. Passive, we vaccinate you with like I got the flu shot recently, and they said they gave me a killed virus, so I'll never get the flu from that. Sometimes if you get an attenuated virus, you might get a flu, okay, because it's just a weekend. And then a passive, you're not making anything here. Naturally, you're receiving through the breast milk or through the, the fetus, in the umbilical cord, okay? And then artificial, like when you get bitten by a rabid dog or whatever, fox, you get jabs for a long time of antibodies for rabies. Because we don't produce antibodies for rabies. Why? Because it's a disease that doesn't affect us. It only affects dogs. Okay? So we have no way of exciting our immune system against that. So we just get antibodies. Okay? So know about that and we're done. Thank you, ladies and gentlemen. I'll give you 5. I'll meet you at 5 past 10, okay? I appreciate it.